Good day. I am Alex De Leon. I will present our paper entitled Supplementation of Broiler Diets with a Multi-Protease Enzyme Increases Growth Performance and Nutrient Digestion of Broiler Chickens. I will be presenting this paper on behalf of the team uh, composed of Alex Wu, Dr. Hans Lee, and Christoph Bodenreider from Chemin Asia, and also Dr. Yo from Changnam National University. Although soybean meal is a highly digestible raw material, it contains anti-nutritional factors. These anti-nutritional factors, if they are not controlled in formula, can result to decrease in performance, resulting from a decrease in the digestion of proteins, or can cause intestinal damage and may lead to impairment of the immune system. Exogenous proteases can help manage these ANFs and reduce the negative effects in performance. This study was done to investigate the effect of the multi-protease enzyme on the productivity of broilers under reduced crude protein and amino acid diet conditions. The experiment was conducted at the Department of Animal Science and Biotechnology at Changnam National University in Korea. The experiment used about 504 birds in 9 treatments. Each treatment was replicated 8 times. In order to create the treatment diets, two basal diets were created, one for 1 to 21 days and another for 22 to 35 days. The treatment diet design included a positive control, a negative control one, which has a 0.5% lower CP compared to positive control and 2% lower digestible amino acid compared to the positive control. The amino acid that were lower down was lysine, methionine plus cysteine, and threonine. Negative control 2, which is 1% lower in CP and 4% lower in digestible amino acid compared to positive control. Again, the amino acid that were lower down was lysine, the total sulfur amino acid, methionine and cysteine, and threonine. From these three, we created the 9 treatment diets, a positive control, and a positive control with 150 grams per ton protease inclusion, a negative control one with either a 150 grams per ton or 300 grams per ton inclusion of the multi-protease enzyme, the NC2 or negative control 2 with either 150 grams, 200 grams, or 300 grams per ton inclusion of the multi-protease enzyme. All treatment diets were provided ad libitum in a mash form until 35 days of age, just to counter check all the amino acids compared to formulated and actual. The method described by Cohen in 2001 was used. Also, we use chromium oxide powder as the internal indigestible marker for the digestibility analysis for part of the trial. For the parameters that were measured, weekly body weights were measured, weekly feed consumption was noted. From this, the average daily gain, average daily feed intake, and feed conversion ratios were computed. For the ileal digestibility, digesta samples from the ileum were collected for analysis of dry matter, crude protein, ether extract, and for the computation for gross energy. The ileum samples were taken at 35 days. About 1 centimeter piece of the ileum was taken after the birds were sacrificed. The digestibility coefficient of nutrients was calculated as described by Wang in 2005. For the results, this table is showing the effects of protease on growth performance of broilers for 1 to 21 days, 28 to 35 days, and looking at the performance from start of the experiment to 35 days. The positive control group with the on-top application of 150 grams per ton yielded the best performance for 1 to 21 days, 28 to 35 days, and for the whole duration of the experiment. Noting that ADG, total gain, and FCR is best for this treatment group among all treatment groups. For 1 to 21 days, it can be noted that although there was no statistical differences in the performance, addition of the multi-protease enzyme yielded increase in performance. For 28 to 35 days, there is significant differences in ADG. I want you to focus on the group having 1% lower CP and 4% lower amino acid. At 200 grams per ton supplementation of the multi-protease enzyme, it was able to recover the loss of CP and nutrients from the lowered CP and amino acids. It is the same picture when you look at 1 to 35 days performance. The group the group of 1% lower CP and 4% lower amino acids were able to compensate for the lost performance with 200 grams per ton of supplementation of the multi-protease enzyme. This actually coincides also with the observation done by by an experiment done by Angel in 2011, where they also lowered down the crude protein of the diet, and at 200 grams per ton, they were able to compensate for the lost performance of the birds. This next table is showing the effect of the protease in diet on ileal digestibility coefficient of dry matter, crude protein, and energy in broilers. There were significant improvements observed in the ileal digestibility of dry matter, CP, and energy with supplementation of protease. The protease improved energy digestibility as a cumulative effect of increased protein digestibility. In hindsight, in animal performance parameters, again, application of protease to the control diets result to numerically higher performance at 35 days. At 35 days, the protease could compensate performance with 1% lowered CP and 4% lowered amino acid. This is at the 200 grams per ton inclusion of the protease. In ileal digestibility, there were significant improvements observed in the ileal digestibility of dry matter, CP, and energy with supplementation of protease. The protease improvements in energy digestibility was as a result of the cumulative effect of increased protein digestibility. In conclusion, in the experiment, the addition of protease had significant benefits on the performance, mainly by increasing the digestibility of CP and consequently digestible energy. Results indicated that the supplementation of protease into the reduced CP and amino acid diets can improve the growth performance of broiler chickens. Thank you very much.